The overall goal of this procedure is to assess expression levels of all genes in samples from electroporated chick embryonic spinal cords. This is accomplished by first in ovo electroporating the right side of early embryonic neural tubes and re-incubating embryos to the desired stage. The second step of the procedure is to dissect successfully transfected neural tube halves in an RNase-free environment and proceed to lysis as quickly as possible. The third step is to isolate RNA and subject the samples to library preparation and high-throughput sequencing. The final step is to process the datasets to remove sequencing artifacts, map reads to the genome, and assign expression levels to each gene. Ultimately, results can show differentially expressed genes through comparison of expression levels in control and experimental conditions.
Visual demonstration of this method is critical, as the neural tube dissection steps are difficult to learn because they require familiarity with embryo manipulation.
For this demonstration, the embryos have been transformed with a GFP-expressing construct at the neural tube. Prepare to spend 2 hours working on up to 10 embryos to collect 1 to 2 micrograms of total RNA per embryo. Under RNase-free conditions, harvest the embryos using fine dissecting scissors and a sieve collection spoon. Transfer each embryo to cold DEPC-treated PBS. Use the solution for all of the neural tube isolation steps.